Hello students, this is Dr. Ashwat Narayan. In this session, we're going to discuss about transgenic approaches in plants for fertilizer management. So you already know that plant need the crucial minerals fertilizer like NPK for the growth and development and produce the more yield, right? So because of that, instead of giving NPK that will cause a soil erosion and then the beneficial microorganisms will die because of the uh, inoculation of the chemical fertilizer. So we need to go with a transgenic approaches. Different approaches are there. We're going to discuss one by one so that these transgenic plant will cope up the scarcity of the fertilizer or nutrients in the soil. Even with that transgenic plant will grow in a soil, even single nutrient is not available. So like that, different approaches are there. We're gonna discuss one by one here. The first one is, we can go with the a nitrogenous gene, that fixes the atmospheric nitrogen here. So there is a different way of nitrogen fixation will be there, like physical fixation by lightning, thundering, or non-symbiotic nitrogen fixation by different types of bacteria, aerobic bacteria like azotobacter, and mycobacterium, arthrobacter, and bacillus that will freely available in the soil, and then they fixes atmospheric nitrogen because of the chemoautotrophic nature. Even some symbiotic bacteria will be there. They will, like a rhizobium, they will symbiotically associated with the plant, like leguminous plants, and then fix the atmospheric nitrogen. And these are the strategies we can use in cope up the fertilizer need in the plants here. So the first strategy is the introduction of the nitrogen fixing gene and associate gene into the agriculture crops. So this is very important, and we can use this strategy in nitrogen fixation so that all agriculture crops will be genetically modified in such a way that they contains NIF gene, NOD gene, and HUB gene. So they fixes the atmospheric nitrogen. And other strategies are there, like nitrogen use efficiency, improvement of that, phosphorus usage efficiency, potassium usage efficiency. So different strategies are used. We'll go one by one. The first one is called as NIF gene introduction into the agriculture crops. What is NIF gene, and what is the strategy we can use it? We'll discuss in the next slide. So you need to know what is NIF gene, where it is present. Though NIF genes and other genes, associated genes that fixes atmospheric nitrogen in symbiotic leguminous plants by action of a bacterium called as rhizobium rhizogene bacteria. Rhizobium rhizogen is a symbiotic bacteria that is present in the soil, but because of the chemotoxic tactic movements, rhizobium, because whenever wound happens, whenever some plants lacks the nitrogen, they will secrete a chemical called as epigenin and luteolin. These epigenin and luteolin will be secreted. These are the flavonoid compounds which produce from the roots of leguminous plants that suffer from the scarcity of nitrogen. So these rhizobium bacteria that is in the soil, because of the chemotactic movement that is produced from the flavonoids like epigenin and luteolin, these chemicals released out from the roots of the nitrogen scarce, the plants which suffering from the scarcity of nitrogen will secrete this, and rhizobium bacteria will comes to the root nodule, and then because they contain three genes. They contain three genes, and the first one is called as NOD genes, second one is called as NIF genes, and third one is called as HUB genes. They will integrate into the plant, and then they forms root nodule. And the rhizobium, because of the presence of these genes, NIF genes, NOD genes, HUB genes, they will have different role in nitrogen fixation. If you take NOD gene mainly responsible for the node formation in the root nodule of leguminous plant. Next one is called as NIF gene. This is the true gene which fixes the atmospheric nitrogen and converts into the ammonia. And then hub genes are there present. You already know that ammonia is toxic to plants, animals, and other animals too, and even the aquatic animals too. Because of that, ammonia should be neutralized and then should convert into amino acids. The up genes will take part in the nitrogen uptake and then helps in the conversion of these ammonia into amino acids. So in locally that will be produced because the ammonia, if the ammonia is percolate in entire cell, what happens that become toxic. So because of that, locally this amino acid, so ammonia will be converted into amino acid. Nitrogen will be converted into ammonia, and ammonia will be converted into amino acid locally in the roots itself. What's the role here? The nod genes which code for a protein called as nodelin. The nodelin protein, what happens, they will form a knot-like structure, and because of the nodelin gene, the plant will produces a hemoglobin, a special type of hemoglobin called as leg hemoglobin. Remember, right? And NIF gene will produces a enzyme called as nitrogenase. Nitrogenase enzyme which fixes the atmospheric nitrogen. What's the process that will happen here? Whenever nitrogenase will after formation of symbiotic association, what happens? Nitrogenase will fixes the atmospheric nitrogen. The nitrogen will be fixed out, and then if you observe that, this will converts nitrogenase will converts the atmospheric nitrogen into dinitrogen ion. Dinitrogen ion to converts to ammonia, it needs the hydrogen proton, right? To drive the proton, the energy will be drained from the glucose molecule C6H12O6 is there. From this glucose, this proton will be donated, and then this dinitrogen ion will convert into hydrogen because of two hydro proton donation. Again, one more proton donation will converts that into diamide. Again, diamide will convert into the ammonia, and by utilizing the energy proton from the glucose here. And then what happens? The nitrogenase enzymes only fixes the atmospheric nitrogen and converts into dinitrogen in anaerobic condition only, right? In anaerobic condition only, this nitrogen will be fixed by nitrogenase enzyme by the NIF genes and then converts into dinitrogen. So to create anaerobic environment, already we discussed about the NOD gene, which because of the symbiotic association, the leguminosi plants produces a special type of hemoglobin called as leg hemoglobin. This leg hemoglobin, what's the role of leg hemoglobin means? It will scavenge the extra oxygen in the root nodule and create a hypoxic condition. Hypoxic condition means oxygen concentration will be neutralized, so that the nitrogenase enzyme will fixes the atmospheric nitrogen and converts into the dinitrogen. Dinitrogen will be converted in anaerobic condition only, and to create anaerobic condition, the leg hemoglobin produced from the plant will scavenge the oxygen and then create a hypoxic condition or anaerobic condition, and this way the nitrogenase enzyme from the NIF genes will fixes the atmospheric nitrogen. And ammonia produced by nitrogen it immediately converts into ammonium ion, and ammonium ion or ammonia will be toxic to plant, again converts into the amino acid locally in the site of generation. Locally in the same cell only, it will convert into amino acid, and the amino acid will be formation of amino acid will be done by different mechanism called as reactive amination, catalytic amination, and transamination process, so that the amino acids will be in the cell site of synthesis of ammonia, they will generate the amino acid. So likewise, the NIF gene and other associated genes will take part. NIF gene, NOD gene, hub gene will take part in the fixation of atmospheric nitrogen and converts into amino acid. And how you engineer the NIF genes into the non-leguminosy plants to get the nitrogen fixation. Here, three genes are necessary. We need to have NOD genes, we need to have NIF genes, and hub genes should be integrated. NOD gene will have a cluster here. We can see NOD A, B, C, D up to J will be there, and node I is very important. It helps in the transportation of the whatever the things that helps in the nodule formation in the symbiotic association. NIF genes is also a cluster. It it ranges from HDK up to Q is present, A, B, C, D, and here if L and A is very important, that produces nitrogenase enzyme. So because of that, NIF gene designing, not gene designing is very important. In we can introduce directly into the chloroplast instead of the nuclear genome. Why? Because chloroplast genome contains a unnecessary plant. We can remove that and integrates directly by gene gun method or by agrobacterium efficient gene transfer here, right? And then later on, we need to go and focus on the vector designing and codon optimization. Why? Because vector should be maybe TI plasmid we can use, or we can use PBR 322 PUC 19 in designing the promoter. And then we need to have a strong promoter like uh, we need to have a strong promoter. Tobacco mosaic virus, strong promoter, 35S promoter will be there, that can be used for the proper expression. And one more thing is, we need to optimize the codon. Why? Because here the NIF genes, not gene, mainly originate from the bacteria. Bacteria doesn't contains any non-coding regions. Intervening sequences will not be there, right? Intron will be not be there. So because of that, codon optimization will be there. Why? Because we are inserting into the plants. In plants, the genes, the DNA should go for replication and transcription. In transcription, what happens? So after transcription modification, the capping and tiling and splicing will be there. If splicing will occur, definitely, if we truly insert only coding sequence, definitely there will be splicing of the genes will happen, and then there is no expression of the NIF gene will happen. Nitrogenase enzyme will not produce, because there is a incompatibility of genes with respect to bacteria and then plants. Plants will have intervening sequence and exons that will be spliced out in post transcription modification. Because of that, we need to go for codon optimization to enhance the translation efficiency in the host plant. And transformation can be achieved by two methods: indirect method by agrobacterium tumefaciens, or direct method by protoplast fusion. Biolistic methods can be used, and then we can select, and we can select the plant, a hybridized plant. Like that, we can insert the NIF genes, and then we can get the nitrogen fixation in non-leguminous plants. So second strategy is, we can use a nitrogen use efficiency strategy in non-leguminous plants. You already know that NIF genes are introduced, and then that fixes the atmospheric nitrogen. We'll have a clusters of other nitrogen fixing genes out there. Those genes in different bacterium will be amalgamated in a single plant, so that nitrogen uptake effectively, where the plant will genetically modified plant can take up the nitrogen from the atmosphere with different genes here. These are the genes that is mentioned, that fixes the atmospheric nitrogen. We'll take that into single plant, so that effectively nitrogen fixation will happen. Along with nitrogen, we need to have NPK, right? Nitrogen, phosphorus, and potash. So here, phosphorus uptake can be achieved by different genes here. These are the genes, and even we can go with a beneficial bacterium or other type of fungus uh, that fixes the phosphorus into the plants here. We'll go with genetic engineering technology, as well as the other type of technology, like beneficial bacterium that is modified in such a way that fixes the phosphorus into the plant cell. So these are the genes like PHOA, PHOB, and PHN genes that fixes the phosphorus in the soil into the plants here. These are the phosphorus uptaking genes. So we can go with genetic engineering technology. We'll flank with a strong promoter, and because of the expression, we'll get the phosphorus fixation more in numbers in genetically modified plants. Even we'll go with the potassium efficiency, where some genes are there that fixes the potassium. Here HAK, KUP, KT family, AKT1, TBL, HKT, KEA families are there. These family of genes mainly helps in the potassium transportation and uptake from the soil, so that if you engineer these genes into the genetic a transgenic plant, definitely the uptake potassium uptake capability of the transgenic plant will be double folded. Even we can go for one more strategy called as symbiotic nitrogen fixation. Other than rhizobium, many other type of rhizobium subspecies will be there. They are specific to soybean, other type of legume plants. So we'll go and then staking all together in a single plant. By genetic engineering technology, we can collect all the genes and then fix that into a single transgenic gene that will effectively fixes the atmospheric nitrogen. Even we can take use of beneficial microbial interaction where other non-symbiotic soil bacterium is there. We'll modify these bacterium in such a way that they will produce multiple folds of nitrogen in their cell, and then and even they will enrich the soil. In the soil, these are the azotobacter, clostridium, klebsiella, cyanobacterium, azospirillum. These bacteria mainly free living in the soil. They will fix us the rhizosphere, soil sphere, and then nitrogen will be more because of genetic modifications here. And one more strategy we can use here is nutrient biofortification. Nutrient biofortification means through genetic modification we can increase the level of essential nutrient in the uh, different plant part of the plants. You can see here ferritin, NAS, IRT1. These are the genes mainly helps in. If they are overexpressed, they will accumulate more number of iron, other type of nutrients in the plant cell itself. Because of more accumulation, there is no need of soil micro minerals and other things that should uptake. Plants itself produces these accumulation of these micro sorry, minerals because of over expression of these genes. So here, in biofortification program, that means enrichment of the nutrients in the edible plants will go with these genes, and these genes mainly helps in the over uptake of the metal ions into the plant cell. One more strategy we can use it in transgenic plants is abiotic stress tolerance. Things here, whenever whenever nutritional uptake is less, then that will cause a stress in plants. Take example in nitrogen and phosphorus unavailability in the soil, the plant will suffer from necrosis, chlorosis, etc. Instead of that, what happens? We will enhances will enhance will modify a plant in such a way that they produces these genes overexpress, and then that can cope up the less amount of nitrogen and phosphorus here. That means some proteins are there. These proteins mainly combat physical stress and stabilize the protein, stabilize the nutrients in this inside the cell itself without the need of the extra soil minerals here. Even we can grow these genetically engineered plants in a barren land without soil even though that doesn't contains any nutrients at all. If you engineer these genes into the plants, it will easily cope up the stress, abiotic stress that is created because of nutritional deficiency. And one more strategy we can use it: enhance nutrient mobilization and remobilization. So as we discussed, some genes helps in nutritional uptake, but some protein family also helps in the transportation of these nutrients into different plant pods. You can leaves, stems. These proteins mainly helps in the transportation of these minerals and proteins effectively in different plant pods, like nitrate transporters, AMT, ammonium transporter, HKT, iron regulated transporters that helps in the transportation of these minerals effectively and distribute throughout in the plant pot. So in that, if you engineer these genes, what happens? That will enhances the efflux of these nutrients in entire plant pot, and then there is no deficiency that takes place that will manage the fertilizer demand. And last technology we can discuss here is called genome editing technology. Will modify specifically the plant genome without without insertion of any genes, foreign genes, and creating GMO. We can precisely edit the plant, stress or nutritional uptake in such a way that we will edit a gene by CRISPR-Cas9 or talent technology or zinc finger technology that can manipulate the plant genome that effectively cope up the stress and uptake the nutrients and reduce the environmental impact and then improve the crop productivity. Like that, we can enhance, we can introduce many strategies by using genome editing technology in fertilizer management in transgenic plants. These are the strategies we are discussed regarding the management, fertilizer management in transgenic plants, so that easily cope up the deficiency of the nutrients in the soil itself.